Hello, and welcome to this e-poster session on autoinflammation and autoimmunity. I am Fabio Candotti, and I'm chairing this section together with Stefano Volpi, my co-chair. Just for the audience, the presenters will have 5 minutes to present their poster, and then they will have 2 minutes for questions and answers. If possible, please pose your questions through the question and answer chat, but also you have the possibility to raise your hand and speak your question directly. I don't think I have anything else to say. I'm just going to introduce the first speaker, who is Valentina Strohmeier, and she will be talking to us about decoding the mechanisms of common variable immunodeficiency associated enteropathy. Valentina, please.
Okay, thank you. Good evening, everyone. I'm a PhD student at Klaus Warnatz's lab in Freiburg, and we work with common variable immunodeficiency patients. And my project focuses on CVID enteropathy, which affects the small intestine, more closely the duodenum, and it depicts as villus atrophy, similar to celiac disease, with regard to disease pathology, but not to etiology, since our patients do not respond to a gluten-free diet. We analyzed now more than 100 patients in our Freiburg CVID gastrointestinal cohort, and we analyzed more closely 30 patients by transcriptome analysis, by histology, and by immune cell phenotyping. In the lower figure, you can see clinical manifestations among the 3 different groups, which are CVID villus atrophy patients, CVID no villus atrophy patients, which still suffer from gastrointestinal complaints and inflammation, but which do not show villus atrophy, and healthy controls, and one clinical manifestation that negatively correlated with the presence of villus atrophy is the presence of plasma cells, and more specifically also IgA positive plasma cells. In this graph, you can see an H&E staining of a healthy control villi with a nice villi shape, and in the middle of a CVID duodenum of a CVID no villus atrophy patient, and the arrowheads indicate some mild inflammation and apoptotic bodies. And on the right-hand side, you can see a villus atrophy duodenum with a severe villus atrophy and complete loss of the apical structure of the villi. Next, we addressed also if there was an increased expression of gamma interferon within the tissue. And on the right-hand side, you can see a high interferon signal, interferon gamma expression in the CVID VA duodenum, and elevated interferon gamma expression shown in brown in the CVID no VA duodenum compared to healthy controls. This upregulated interferon gamma expression in the villus atrophy patient was shown to be significant compared to healthy controls. Next, we also analyzed the transcriptome of the 3 groups by bulk RNA sequencing, and in the principal component analysis plot, you can see the nice clustering of all the 3 groups. And by looking at the differentially expressed genes in the Venn diagram, we found many up and down regulated genes by comparing the 3 different groups. And interestingly, by comparing CVID VA compared to CVID no VA tissue, we found only 7 upregulated genes and more than 70 downregulated genes, and many of those downregulated genes in villus atrophy were addressing or were involved in tissue homeostasis as mucus genes, MAC2, MAC6, or aquaporins, and this makes complete sense due to the tissue destruction in villus atrophy. More interestingly, we observed also a highly increased interferon signature, here shown as a heat map of interferon response genes, in red upregulated expression and in blue downregulated expression. On the top part of the figure, you find some information about the tissue donors as norovirus infection, the presence of IgA positive plasma cells, and the Marsh scoring, which is a scoring used in celiac disease to describe the severity of tissue inflammation and destruction. And we also double-checked 4 of those genes by qPCR, and those genes were published as biomarkers or potentially biomarkers in celiac disease. And we also observed a significant upregulation for interferon gamma, IFI27, and GPP5 in the villus atrophy group compared to healthy controls. Next, we were also interested in the effect of norovirus infection on the interferon response gene expression. And indicated by the blue arrows, you see patients with a norovirus infection, and you can see that they had even a more increased interferon signature. And we also double-checked this by qPCR. In red dots shown villus atrophy patients, and in blue no villus atrophy patients, but grouped by norovirus status. And all those 4 genes involved also in antiviral response as OS2 were significantly upregulated in tissues that had norovirus infection. Furthermore, we were also interested in the immune cells that would accumulate in the atrophos villi. And on the right-hand side, you can see a CVID villus atrophy duodenum with infiltrates, and most of the infiltrates were CD163 positive macrophages, here shown in red, together with CD8 positive T cells, shown in green, and those T cells were often also expressing granzyme B, shown in white. And these cells were also present in CVID no-VA patients, but there they localized more to the subepithelial layer. And we addressed also the phenotype of these macrophages accumulating in villus atrophy, and gated them by HLA-DR positive to DIM expression and CD14 positive expression, and further subdivided them into CD11C negative, more homeostatic macrophages, and CD11C positive, more inflammatory-like macrophages. And this these macrophages were significantly expanded in villus atrophy compared to healthy controls. This was my last figure, and I'm happy to take your questions.
Thank you, Valentina. There is 1 minute left for a question, quick question for anybody else. Would JAK inhibitors have a role in this pathology?
I mean, I did not analyze patients on JAK inhibitors, but due to the high interferon signature, it might make sense, yes.
Then in the clinical features, I see there was very little that was specifically related to the patients with the VA.
Yeah.
There was nothing, there was only them.
Sorry, the last part.
Almost nothing that was only present in the patients with the VA. Most of these clinical features were in common with other subgroups. Yeah. This is like one problem that we're facing, because CVID itself has a strong effect. And then there comes also this villus atrophy, but which only affects a part of the intestinal organs, of course. And CVID itself has also a high impact on the tissue state in the duodenum. That's actually what we believe, but we're still eager to find out what makes the difference. Okay, thank you, Valentina.
You're welcome.
Thank you very much. So we welcome the next speaker. There is Ronan Leahy, and we'll talk about an identification of an NLRP6 mutation in a patient with autoinflammatory disease.
So thank you. I'd like to thank the organizers for allowing me to present this work, which, as you mentioned, relates to the identification of an NLRP6 mutation in a child who presented with autoinflammatory disease. The work was done in Dr. Emma Cray's lab in Trinity College Dublin by Dr. Sinead Kennelly in collaboration with our colleagues in UCL, in particular Dr. Helen Lachman. I'm the clinician, one of the clinicians looking after the patient. So, okay, can you see now? Is that okay? Yeah. So sorry about that. I'll just move on with the clinical phenotype. So the patient first presented to us at the age of 8, but the mum reported that the child had actually had painful nodular swellings on her limbs from infancy. You can see here from the panel here on the top left. She also had anorexia and iron deficiency anemia. So we did an endoscopy which demonstrated some macroscopic changes of inflammatory bowel disease, but she also had transaminitis. And as you can see from the photomicrograph on the top right, she had a lobular hepatitis with some granulomata as well. So ultimately, we tried a combination of different treatments, but finally got a good clinical and biochemical response to adalimumab. And you can see here in the panel that she got a dose-dependent response to adalimumab, and she's still on this treatment subcutaneously once a week. So thanks to our collaboration with Dr. Lachman, we were able to enroll the patient on a next-generation sequencing study undertaken in UCL, where using the VIP panel, they examined the sequencing of 166 genes associated with inflammation and vasculitis. And she had 4 mutations, 3 of which we now know are clinically irrelevant, but she was found to be heterozygous for an R653G mutation in the NLRP6 gene. So this mutation is between leucine-rich repeat region 1 and 2. It's on the external surface of the gene, of the protein, in a functionally important part of the gene, and is predicted by polyphen to be probably pathogenic. So we thought this mutation might be important, and we wanted to explore it a little bit further. So NLRP6 works in 2 ways. One way it works is it regulates the NF-kappa B pathway. So we introduced, with site-directed mutagenesis, we introduced the mutant protein into HEK293 cells. And you can see here in the panel on the left that those cells that have the mutant protein are unable to regulate an expression of NF-kappa B. Then we also compared peripheral blood mononuclear cells from the patient against healthy controls, and you can see evidence of upregulation of NF-kappa B pathway with early degradation of the I-kappa beta protein and increased and earlier expression of caspase-1, pro-IL-1 beta. And also you can see increased and earlier expression of phospho-ERK, suggesting increased activation of the MAP kinase pathway as well. So in addition to its effect on the NF-kappa B pathway, the NLRP6 protein is also involved in inflammasome formation. So we examined this. On the left, you can see confocal microscopy comparing ASC specs in cells that have the mutant protein versus cells that don't. And you can see that there isn't any difference in the presence of aspects. So no evidence of an effect on the inflammasome produced by the mutant protein. We also collected peripheral blood mononuclear cells from the patient, and again from the 3 healthy controls, and we stimulated the cells with heat-killed gram-positive and gram-negative bacteria, and measured both interleukin-1 beta and interleukin-18 production over time as a measure of inflammasome activity. And again, comparing the NLRP6 mutant protein PBMCs versus the controls, we saw no difference. So again, suggesting that there was the protein, the mutated protein, didn't in any way affect inflammasome production in the peripheral blood mononuclear cells. So because I only have 5 minutes, I'll just summarize very quickly. This is a patient, she's now an adult, who presented in infancy or with symptoms of autoinflammatory disease, and was found to carry a mutation in NLRP6. She developed a good response to anti-TNF alpha treatment. Functional studies appeared to show no effect on inflammasome formation, but apparent upregulation of the NF-kappa B pathway. Now, curiously enough, the patient's mother also carries the mutation, and she's asymptomatic. So it's a bit difficult at this point to say whether this is an association or whether it is causative. And then no other patients to date with mutations in NLRP6 have presented with autoinflammatory disease. So we're planning to do further genetic work. In other words, we're going to do an exome on the patient to make sure there is no other molecular basis for her condition. And we may undertake some functional work on mum as well to see whether she has functional phenotype to go with the mutation. So sorry about the technical issues. I'm very happy to take any questions at this point.
Thank you very much. So we have a question from Bodo Grimbacher. There were 2 other mutations in CCR1, apparently. So why this is not a DADA2 patient also because he responded to anti-TNF treatments?
Sure. So we measured the ADA2 enzyme activity and it appears normal. So that's what we were thinking first. And in fact, that was what was suggested in the original manuscript. So it's a very good question, but we're happy that the enzyme activity is normal, assuming that's a good readout of ADA2 activity.
And another question is if you check expression of NLRP6 in patient cells, or if you get any modification in the mRNA of IL-1 beta or IL-18?
So I think it was, I didn't do all the experiments myself, no, but I think it was mainly Western blot that we used rather than mRNA to demonstrate the effect.
Okay, I have a question. Was it a de novo mutation or was it?
Yeah, I mentioned, So I suppose we have a number of, we're slightly skeptical about this ourselves because the patient's mother also carries this mutation and she doesn't appear to have any clinical symptoms. But of course, you can often see incomplete penetrance in autoinflammatory disease. So we're keeping an open mind.
Okay. Indeed, this is one of the problem that we often incur to find this mutation and to giving it a role. Okay, thank you. Thank you very much for your
Thank you very much. Thank you.
Okay, thank you. We'll go to the next speaker, who's Mate Krausz, and he will tell us about the microbiome signatures in patients with CTLA-4 insufficiency. Mate, please.
So welcome, I will talk about the microbiome in CTLA-4 deficiency. CTLA-4 is an essential player in the regulation of the immune responses. It is an antagonist of CD28 and outcompetes it by binding the ligands CD80 and 86 with a higher affinity. Heterozygous germline mutations in humans cause CTLA-4 deficiency, which is a disease which has patients with the disease have severe immunodeficiency and also immune dysregulations. Main clinical manifestations include autoimmune cytopenia, lymphoproliferation, hypogammaglobulinemia, and as well respiratory, intestinal, neurological, and skin involvement. In the largest currently published cohort, which includes more than 200 individuals, the clinical penetrance is around 70%. So we have the questions, what determines the penetrance and the expressivity of the disease? To approach this, we divided CTLA-4 mutations carriers into 2 groups, affected and unaffected mutation carriers, and collected samples from them. In addition, we also hypothesized that the intestinal microbiome may identify patients with mutation in CTLA-4 and a history of enteropathy. We collected stabilized stool samples from these patients and household controls, altogether 17 affected, 9 unaffected, and 28 control samples. Did fecal DNA extraction and performed a standard Illumina MI6 sequencing of the 16S ribosomal RNA sequence. We combined the sequencing data with clinical data sets and a deep questionnaire about lifestyle, eating habits, and self-assessment of the disease. To come to the results, we analyzed the alpha diversity in these 3 subgroups, and strikingly we found that affected CTLA-4 mutation carriers had a significantly decreased Shannon diversity index compared to unaffected or wild type, which means the healthy controls. it was also really interesting for us that there was no significant difference in the diversity index between unaffected mutation carriers and the wild-type persons. This was further confirmed by the beta diversity analysis. As you may see, the affected carriers cluster together separately from the unaffected and healthy individuals. To go on, we compared again the alpha diversity in CTLA4 mutation carriers with or without the history of enteropathy. And we found that patients with reported enteropathy have a tendentially lower alpha diversity compared to ones without. To go on with the beta diversity, this was also again confirmed as the patients with reported enteropathy clustered together separately from the ones without enteropathy. Looking at the species, we find significantly upregulated or depleted bacterial species, in altogether 10 taxonomical levels, and some of them are already reported in IBD, inflammatory bowel disease, and with the same phenotype, so significantly decreased or enriched. To sum up our findings, we found that the affected CTLA4 mutation carriers had significantly decreased alpha diversity compared to unaffected carriers and healthy controls as well. The beta diversity confirmed these results as the structure of the microbiome in affected carriers was different from the unaffected mutation carriers or healthy controls. The change in this gut microbiota therefore could be a disease modifier in the CTLA4 deficiency. Regarding the enteropathy, we found that the mutation carriers with history of enteropathy have a distinct signature that is some part similar to the microbiome reported in inflammatory bowel disease. These identified taxa that were significantly upregulated or depleted in the gut might be future potential therapeutic targets. Thank you.
Thank you. See if you have any questions. Not yet. Were the CTLA4 patients on immune suppression?
These patients are the regulars at our clinic, and we did not cluster them with the therapy. These samples were collected some time ago, so these patients were not on the, for example, abatacept, but basic immune suppression. I would say yes.
And then the question that I share with Bodo is this an egg or a chicken problem? Is the enteropathy coming first, it changes the microbiome, or is the microbiome changes that cause the enteropathy?
It is a very good question. We are also really interested in that. We are trying to answer this question in the coming years, more or less, and we will use so we follow up the unaffected patients, and in some data that is not shown here, we could already identify with the functional assessment of these the microbial pathways, that some pathways, so metabolic pathways, are altered in the unaffected ones, which could be interesting to see how the unaffected patients develop. And on the other side, we are now currently analyzing more samples in longitudinal with or without treatment, and especially looking at treatment with abatacept, which is a specific treatment for CTLA4 deficiency. And this might answer a bit about this question.
And what about the use of antibiotics in your patients?
So in these kind of questions, they were excluded from this cohort, as far as I remember.
Okay, I think that's all we have time for. Thank you, Martin.
Thank you.
Okay, the next presenter is Krausz Berbers, talking about multiplex proteomic that can classify inflammatory complication in common variable immunodeficiency.
Yes, hi everyone. Can you hear me? I think so. So my name is Rose, and I'd like to share with you our data on targeted proteomics in immune dysregulation in CVID. This was work done by me and Helen Leavis with collaboration of the Erasmus MC in Rotterdam, Groningen, and also Freiburg in Germany. So we wanted to focus for this study on whether we could develop new biomarkers for immune dysregulation CVID patients, because as we all know, these complications cause the most morbidity and mortality in CVID, and it's currently quite difficult to predict which patients can develop immune dysregulation. And also when they have developed it, it's difficult to measure their disease progression. The approach we chose to investigate this was to use a new targeted proteomics technique, which is based on a proximity extension assay. And this allows you to measure many serum cytokines in very small samples. So what we did is we sampled 2 separate cohorts individually. We measured 180 markers in the first cohort, and then in the second cohort, we trained an algorithm on the first cohort and then on these markers, and then use the second cohort to validate how good it was at predicting which patients had immune dysregulation and which ones didn't. So here's a very large overview of the 2 cohorts. So the sample size, the first cohort, we had 15 healthy controls, 16 patients with CVID with infection only, and 14 with CVID and immune dysregulation. This first cohort was age and gender balanced, and the most prevalent clinical phenotype was autoimmunity. And the second cohort was slightly larger, where we had 27 healthy controls, 24 patients with CVID and infection only, and 23 with CVID and immune dysregulation. Now, some of these patients had a known monogenic background. So in the first cohort, we have 1 patient with a CTLA-4 mutation, and in the second cohort, we have 3 TACI mutations and 1 patient with a PI3 kinase mutation. Who, by the way, all these mutations were unknown at time of sampling. This is what I already said. So we measured 180 markers. For the training cohort of the first cohort, we trained a classification algorithm we tried 3 different machine learning algorithms, of which ElasticNet was the best one. And then we tested this on the second independently sampled cohort, and then we see how well it performed. So these are the results of that. So on the second cohort, an algorithm using 5 markers, which are Miller-1, LILRB4, IL-10, IL-12, RB1, and CD83, could predict which patients had immune dysregulation and which ones had infection only, with a sensitivity of 0.83 and a specificity of 0.75. And what's interesting is that in the second cohort, we included 13 patients from Freiburg in Germany, there were no patients from this center in the first cohort. And this algorithm correctly identified 12 out of 13 patients. And if you look at the individual markers, what we see is that IL-10, IL-12, RB1, and CD83 were consistently upregulated also in the testing cohort, while Miller-1 and LILRB4 were not significantly upregulated in the testing cohort, and therefore further studies we would need to validate whether perhaps these 2 can be left out, and we could have the same prediction with IL-10, IL-12, RB1, and CD83. We then pooled both of the cohorts to see whether we could also learn something about the pathogenesis or the underlying immune dysregulation. So here's a volcano plot where on the right we can see CVID with immune dysregulation, and on the left we can see markers upregulated in CVID with infection only. And these were all inflammation-related markers, so we're expecting to see mostly things upregulated in the CVID with immune dysregulation group. So what we see is several markers related to immune regulation, such as IL-10, LAC3, which is an immune checkpoint. We also see TNF-RSF9, which is more commonly known as 4-1BB, and CD83, which is an antigen-presenting cell maturation marker. So this shows that there's a role for immune checkpoint or chronic inflammation markers in immune dysregulation in CVID. And on the other hand, what we see more pro-inflammatory markers. We see, for instance, CXCL9, IL-17, IL-12B, which are more related to a T helper 1, T helper 17 immune response or immune activation. And then if you do a pathway analysis on this, what we see is that the central marker here is the IL-10, which is connected to many of the other proteins that we found upregulated. So to conclude, we have developed an accurate and reproducible tool that can classify patients with immune dysregulation, which uses IL-10, IL-12, RB1, and CD83. Furthermore, we see a possible role for chronic inflammation and perhaps also immune exhaustion in immune dysregulation. And we believe that this might be a first step towards the development of biomarkers that we may use as a clinical tool for monitoring disease progression or perhaps therapeutic response. And I'm very happy to take any questions.
Thank you very much. So I don't see questions so far. I have a question. Congratulations for the design of the study. Do you think this marker might act as a predictor for inflammation? So can you design a longitudinal study, let's say?
Yeah, so that would be the study that you would need to really see if it has proper predictive value. So I should say, based on the study, I cannot answer that question. Of course, we do think that you might see lower levels of inflammation prior to the development of clinical immune dysregulation in these patients. And we would also be interested to see whether they can be used to monitor therapeutic response. So if perhaps you can see resolution of your inflammation when you start immunosuppressive therapy, which would be very useful because often we don't know how well the therapy is working until, well, we need some time.
Okay, thank you very much. We have to go to the next speaker. Thank you.
Okay, thank you. We have in the chat that there might be some technical issues in terms of not seeing the slides turning, but they worked for me, and I'm guessing for Stefan as well.
Yeah, also for me, sorry. I didn't see.
So hopefully there won't be any other issue going forward. The next speaker is Barini Thian, and she will tell us about germline biallelic PI3CG mutations. Barini, please.
Thank you for the introduction. Good evening, everyone. Today I will talk about PIK3CG mutations in a multifaceted immune dysregulation. I have nothing to disclose. We first studied a patient with hemophagocytic lymphohistiocytosis or HLH-like phenotype. Using our patient is characterized by prolonged fever, hemophagocytosis in the bone marrow, and high levels of ferritin and triglyceride levels. Since only 4 out of 8 criteria were met, our patients therefore suffered from a HLH-like disease. Using whole exome sequencing, we found no rare variants, homozygous variants in this patient. However, we identified compound heterozygous variants in PIK3CG encoding for P110 gamma. This is the catalytic subunit of the PI3K gamma complex, in which its expression is highly enriched in immune cells. In figure B, you can see that the amino acid affected are evolutionarily conserved among species. However, the mutant protein is expressed in patient-derived T cells. P110 gamma belongs to class 1B PI3 kinase, which is predominantly activated by G protein couple receptor. Activation of PI3K gamma leads to the generation of lipid second messenger phosphatidylinositol 3 or PIP3. One of the downstream targets of PIP3 is the activation of AKT. We thus hypothesize that a loss of function in PI3K gamma may result in the impairment of AKT activation. Indeed, using SDF1 or CXCL12, which activates GPCR and thus PI3K gamma, we found that patient-derived T cells show a reduction in phosphorylation of AKT upon stimulation compared to cells from healthy donors. To prove causality in an independent cellular system, we created Jurkat PIK3CG knockout cells and found reduced AKT phosphorylation, as you can see in panel B here. And we also see a decrease in activation marker CD69 upregulation. And furthermore, upon reconstitution with wild-type P110 gamma, here you can see that CD69 upregulation on the surface of Jurkat PIK3CG knockout cells. And by contrast, upon reconstitution with mutant vectors here, you can see that there is no upregulation of CD69 was observed compared to empty vector, showing a loss of function mechanisms for both individual variants. PI3K gamma is also highly expressed in myeloid cells and play a role in innate immunity, such as actin rearrangement and phagocytosis. Therefore, we hypothesize that a loss of function in PI3K gamma may result in aberrant actin rearrangement and impairment of phagocytosis. Using immunofluorescence image analysis, we found that patient monocyte-derived macrophages display a dramatic reduction in cell area as well as total amount of F-actin as quantified here in B and C. Consistent to the morphological defect found in patient cells, our patient cells also show a reduction in functional phagocytosis compared to cells from the mother or healthy donor, as can be seen here. In summary, loss of function of PI3K gamma causes HLH-like immune dysregulation with functional defects in T cell and myeloid cells. I'd like to thank the co-authors and my supervisor for their kind support and collaborations. And thank you for your kind attention. I'd like to take questions. Thank you.
Very nice. Thank you, Marini. See if there is any question. Not yet. So were you able to see correction of the actin and the spreading phenotype in the cells, similarly to what you saw when you saw correction in the experiments you did before? Because the monocyte that is isolated is very, very small amount in our patient cells, so we are unable to isolate such a lot of cells from our patients, so we are unable to do that experiment. So we actually did in T cells, so we are able to see the restoration of the CD69 activation in our patient cells. So when we put in this wild type P110 gamma, we are able to restore the phenotype. Okay. But did you do the correction? Yes. So we did it in our patient cells, in T cells. What did you use to correct? So we used the wild type vector to show a rescue in these patient cells ex vivo. So by transfection or with a viral vector? By transfection. Transfection, okay. There are no other questions. Well, thank you again. We'll move to the next presenter.
Thank you very much. The next presentation will be by Ahmet Ozen, and the title is Broadly Effective Metabolic and Immune Recovery with C5 Inhibition in CHAPLE Disease.
Hi, everyone. My name is Ahmet Ozen. I'm a physician at Marmara University in Istanbul. In this talk, I'm going to present the results of a translational study that we carried on a recently discovered human disorder called CHAPLE disease. This study is a product of an overseas collaboration.
Can you start in the presentation form? Because we see all the great, thank you.
So CHAPLE disease is a novel complement disorder caused by genetic loss of CD55, which is a key regulator of the complement system. The cardinal feature of CHAPLE disease is intestinal lymphangiectasia. It causes protein-losing enteropathy and intestinal malabsorption. A second important feature is systemic thrombosis, which may go fatal. When comparing CHAPLE disease with the well-defined complement disorders, paroxysmal nocturnal hemoglobinuria and atypical hemolytic uremic syndrome, all three disorders are caused by insufficient complement regulation, but at different levels. Notably, PNH and aHUS can be effectively treated by the anti-C5 antibody eculizumab. The similarity in the molecular pathways among these three disorders led us to test eculizumab in CHAPLE patients. We treated 16 patients over a period of 20 months, and the clinical efficacy was remarkable. The drug rescued the patients from death, eliminated requirement for albumin transfusion and hospital admission, and reversed all manifestations. The two exceptions to full recovery were thrombosis and thrombocytosis, which were not completely reversed in all cases, as shown here. We carried in-depth explorative studies to better understanding the broad aspects of this disease and how they respond to therapeutic inhibition with C5. CD55 inhibits the C3 and C5 convertases, but eculizumab blocks at the C5 level. Our findings show that terminal complement cascade is inhibited, but upstream branches remain active. This result indicates that C5-mediated injury accounts for most symptoms in CHAPLE, given the therapeutic response. One patient required more frequent eculizumab dosing to control disease activity. We found that he carried a homozygous single nucleotide polymorphism in C5 gene, which is common in the population at a rate of 20%. The isoleucine substitution at position 802 is predicted to cause new clashes upon eculizumab binding. Further investigation revealed that the patient had higher eculizumab C5 complexing and less effective elimination of the circulating form of the membrane attack complex. And we've identified 3 additional patients with the same SNP, which all showed the same response. We further found that 4 patients had elevated serum C4 concentrations. Interestingly, 3 had thrombosis and 1 had persistent thrombocytosis. We carried whole genome sequencing and observed that the C4 gene copy numbers correlated with serum C4 values in our patients. Since CHAPLE disease causes numerous systemic alterations, we hypothesized that the overall proteomic composition may be altered in these patients, and we carried a proteomic profiling using the SomaScan platform that measures 1,300 proteins in the serum. We discovered CHAPLE patients had 94 proteins abnormally regulated at baseline. It was surprising given protein-losing enteropathy that there were more proteins upregulated compared to downregulated ones. Among top functional changes were coagulation and complement systems. Interestingly, the downregulated proteins displayed a better response to treatment as shown in this principal component analysis. 23 out of 26 proteins recovered by 2 weeks. Moreover, a group of proteins with the immunoglobulin-like fold had a significant reduction pre-treatment and showed a striking rebound after treatment. This finding suggests that proteins with this structural property are selectively lost. We know that albumin and immunoglobulins are lost in intestinal lymphangiectasia. Now we discovered that this set of proteins with the Ig-like fold are also selectively lost. Among the biomolecules tested, erythropoietin receptor showed a striking surge following treatment, which was normal at the baseline. This molecule has known functions in the hematopoietic system. It also supports vascular regeneration.
Sorry, 30 seconds left for your presentation.
Okay. Yeah, I'm passing to the last slide. So finally, we assessed the fecal microbiome using shotgun metagenomics, and we found pathological microbiota such as inflammation-associated Enterobacteriaceae and reduced Shannon diversity. There was a greater interpatient dissimilarity of microbiota before treatment and homogenization afterwards. In conclusion, our study shows that CHAPLE disease causes a broad range of pathological changes in immunity, metabolism, and the gut microbiota composition. that are largely reversed by C5 inhibition treatment. Thank you for your patience.
Thank you very much. So, okay, my first question would be, so is this treatment should be prolonged for their lifetime for this patient, correct? So is it a regular treatment? Can you supply enough drug for the patient, since it is a very expensive drug?
Yeah, very nice question, indeed. So it's one of the most expensive drugs in the market. It costs millions of dollars over lifetime. And so, yeah, that's a very good question. And in certain individuals that were inaccessible to treatment over time, we showed that the disease turned back. But there are limited observations in a couple of patients that over a couple of months, when they cessated their treatment, no symptom has come back in the milder cases. But for most subjects, I would say yes, they would require the drug forever.
Okay. So it's the kind of treatment that you can expect to use with the flare. Okay.
Yeah.
Okay. Thank you very much. So we have to go to the next talk for the sake of time. Thank you very much for your talk.
Right. Thank you. The next speaker is Elizabeth Rivers, and she will ask the questions of whether Lewis-Cottaldrich syndrome is a disorder of immunometabolism. Elizabeth.
Thank you very much. So would like to share with you today some work looking at immunometabolism in Wiskott-Aldrich syndrome. So Wiskott-Aldrich syndrome is a severe immunodeficiency disorder resulting from impaired actin cytoskeletal regulation of hematopoietic cells. And an inflammatory phenotype is seen in the vast majority, with features such as severe eczema as shown here on the right, but also including other features such as inflammatory bowel disease, vasculitis and arthritis. So we've shown previously that the Wiskott-Aldrich syndrome protein, or WASP, is important in the autophagy inflammasome axis. And now we can demonstrate a role also for WASP in mitophagy and mitochondrial homeostasis, with important implications for immunometabolism. So first, we looked at mitochondrial homeostasis in primary monocyte-derived macrophages using confocal microscopy. And we found that WASP is essential for maintaining mitochondrial homeostasis. So you can see here mitochondria as highlighted in green. In the healthy, macrophages exist in networks at the basal cell surface. And quite strikingly, what we saw at rest, macrophages from Wiskott patients were quite markedly different in their morphology, showing a much more fragmented appearance. And this morphology was completely restored following clinical gene therapy. And when we inhibit ARP2/3, through which WASP acts in the healthy donor cells, we also find a phenotype that mimics Wiskott, suggesting that the role of WASP in mitochondrial homeostasis is reliant on its actin polymerization functions. So next, we developed a model of inducing mitochondrial damage to look at how mitochondria might be cleared from the cells. So we used a cyanide-containing compound, CCCP, and we found that in healthy donor macrophages, damaged mitochondria became separated from the mitochondrial networks and were entrapped in actin cage-like structures which were targeted for mitophagy. So on the left panel here, we can see in green the mitochondria at a resting state. And upon mitochondrial damage, you see mitochondria become clustered away from the network and become tightly bound by actin in cage-like structures. On the right-hand side, we see that these cage-like structures are also associated with LC3, which is a known autophagy marker. And quite strikingly, we saw in Wiskott patient cells that actin cages were impaired in Wiskott, but restored after clinical gene therapy. And they also appear ARP2/3 dependent. It's also interesting to note that the actin cages that were present in Wiskott cells were much smaller and also did not seem to associate with LC3. And this is interesting because work by others has shown that ineffective clearance of damaged mitochondria is also associated with inflammasome activation. And we wonder if this may go some way to explain some of the other inflammatory features of Wiskott. So finally, we looked to explore some functional aspects of impaired mitochondrial homeostasis in Wiskott. And we found in experiments, seahorse experiments, that Wiskott macrophages exhibited reduced basal mitochondrial respiration. So Wiskott cells shown here in red compared with healthy donors in black. You can see quite a marked reduction in basal mitochondrial respiration. And perhaps even more importantly, we also found that there was reduced maximal respiration, which means that when the cells became stressed, they were unable to meet the metabolic demands of the cell. And when we look back in the literature, we found, interestingly, that other groups had identified very similar metabolic defects in Wiskott platelets as early as 40 to 50 years ago. And we wonder whether if we extend these metabolic defects to other immune cell lineages, we may be able to explain some other immunophenotypic features of Wiskott, including things like T-cell anergy and progressive lymphopenia with age. Thank you, and I'll take any questions.
Thank you very much. Beautiful studies, beautiful pictures. See if I have any questions. I don't see questions. So far you've done these studies in macrophages.
That's correct. Yeah.
That's why that was the initial target cells.
Because our initial work looking at autophagy inflammasome access was also in macrophages. So this study started as an extension of some earlier work looking at autophagy and inflammasome stimulation in non-selective and selective models. So looking at the mitochondria was an extension of that work. We know macrophages are important in the inflammatory response.
Right. And have you saw that you want to do this in other cell lines?
Yes. Yeah.
The T cells or B cells at this point?
Sorry, I just missed that.
Have you looked at all at B cells or T cells at this point?
No, that's very much the focus on the next steps of this work is to extend that to lymphocytes.
Right. I was thinking the same thing, that if you look at cells that rely more on mitochondria, I think regulatory T cells or or memory cells, probably T and B cells, depend on that. Congratulations, beautiful work.
Thank you. Yeah.
There's a question, and Bodo asks if you have any data on Treg numbers and function in Wiskott.
No, we don't have sufficient data to make a comment about that in Treg cell numbers, as far as I'm aware. So in the Wiskott disease, we know that there are normal numbers of Tregs, but a reduced functional activity of those cells. At least if you put them in culture with effector cells, the inhibition of proliferation will be lower in Wiskott cells compared to controls. Absolutely. I would agree. We know that. I don't think that we have data showing that the Treg numbers are reduced in our cohort of Wiskott patients, but I wonder about the function.
Okay.
Thank you very much. So the last speaker is Sinisa Savic, and we'll talk about identification of critical transcriptomic signaling pathways in macrophages from H syndrome patient without autoinflammatory complication.
Right. Well, I'd like to thank the organizers for letting us present some of our work that we've done on H syndrome. I'm presenting this on behalf of my PhD student who couldn't be here today to present the work that he mainly did. So I'll try to do a decent job on his behalf. These are my disclosures. So H syndrome or also known as a histiocytosis lymphadenopathy syndrome, is due to biallelic mutations in a gene called SCLC29A3. This gene codes for a nucleoside transporter, and the effects of the SCLC29A3 deficiency are quite complex on cellular function. So there's lysosomal deficiency, there's also effect on mitochondrial function. Histiocytosis, when it is present, it resembles Rosai-Duffman disease. And there are many non-immunological manifestations of disorder, but up to 25% of patients develop complications which are typically seen in systemic autoinflammatory diseases. Because of this, there is no agreed treatment algorithm for inflammatory complications in this disorder, which has only recently been recognized as autoinflammatory syndrome. So our objective was to investigate which biochemical processes and pathways may be relevant to the pathogenesis of inflammatory complications in H syndrome. So to do this, we focused on investigating monocytes and macrophages because we think these are the most relevant cell types in pathogenesis of these disorders. And furthermore, we selected patients with H syndrome who had no autoinflammatory complications and a patient with both Rosai-Duffman disease as well as autoinflammatory complications. And we also compared the gene expression of these patients to recognize autoinflammatory disorders such as TNF receptor-associated periodic fever syndrome, and we had 2 patients that we used for this comparison. So in Figure A shows heat maps that were generated using a pairwise comparison of gene expression between healthy controls and H2 syndrome patients. HC2 is the one, as I said, with inflammatory complications. And as you can see, the heat map show significant difference between the healthy control and H syndrome patients when you look at the monocytes and one macrophages. Going back. And there's also difference between H1 patient as well as H2, the one with autoinflammatory complications. And then when we looked at the 15 most upregulated and downregulated genes, there was clearly some resemblance between the H1 and H2 patients, but there were also some differences. In Figure D, you can look at the same 15 most upregulated genes in the H syndrome patients as well as the patients with TRAPS. And there are a number of similarly expressed genes between the 2 conditions. So when we then looked at the gene ontology enrichment analysis for differentially expressed genes in M1 macrophages of H syndrome patients and patients with systemic autoinflammatory diseases, there were a number of shared GO terms, including immune response, peptide antigen binding, and interferon signaling pathway, which was of particular interest. We then went on to culture the M1 and M2 macrophages. As you know, the cellular plasticity that confers macrophage capacity to elicit direct immune response, which can be pro-inflammatory with M1 macrophages and anti-inflammatory with M2 macrophages, and this also depends on the environment. And initially what we saw that actually the patients with H syndrome had a proportionally smaller number of M1 macrophages, but when these macrophages were put into the culture, they produced significantly more TNF and also IL-1 beta, and these were comparative levels of TNF production to what the macrophages from the systemic autoinflammatory disease patients with TRAPS were showing. More importantly, also, when we looked at these serum levels of some of these inflammatory cytokines, strikingly, the patient with the H2, both H2 patients secreted quite significant amounts of interferon gamma and also TNF. So in conclusion, regular analysis of monocytes and macrophages subsets in H syndrome shows that these patients have an autoinflammatory phenotype. Regular cytokine analysis shows upregulating interferon gamma pathway which can be selectively targeted these days. And that's just to thank the people who were involved with this project. So I'm happy to take any questions at this point.
Thank you very much. Oh, I don't see questions so far. I have a question. So TRAPS patients are more IL-1 mediated. So this inflammatory phenotype, is it similar to what you see in TRAPS patient or autoinflammatory syndrome, IL-1 mediated ones?
Well, unfortunately, no, because it's interesting because obviously when you look at TRAPS patients transcriptomically, you would suggest they would respond to anti-TNF treatment, but in reality, they never do. And actually, it's the IL-1, obviously, that is the cytokine that is blocking, which really produces good responses. So for the H syndrome, what really prompted the study is because we have a couple of patients with quite difficult inflammatory complications who haven't really responded to under IL-6 inhibition or IL-1 inhibition. So we were looking to see, are there any targets that might be more amenable to treatment? And I think interferon gamma makes quite a lot of sense given the role in macrophage activation. Unfortunately, it's not easy to get the anti-interferon. Ipilimumab is not easily accessible for this, but it would make sense to use.
Yeah, I agree. I don't see.
Okay.
Which was the stimulant? Sorry, you stimulated monocyte with what?
No, this was just a spontaneous production of cytokines in the culture.
Okay.
Just typically, once you differentiate the monocytes using the technique we did, they secrete spontaneously cytokines. So this was just a spontaneous secretion of the TNF in the culture.
Okay, thank you.
Unfortunately, we are running late and we have to close here. I want to congratulate all the presenters. A very interesting session, and I wish you all a good evening. Bye.
Goodbye.
Bye.
Okay.
Thank you.